Thank you everyone for joining today's webinar, Data-Backed Trial Design, Applying AI to Clinical Outcome Analysis. Again, apologies for my camera not working today, but you can see me here. So again, my name is Nick Brown. I'm a member of the Satara AI team. We've been developing and applying advanced deep learning and large language models to use cases across the drug development lifecycle for over the last 7 years. And this has included work from small compound analytics and discovery to clinical trial research and analysis and regulatory approval as well. And as part of that, today, we're going to focus largely on applying these type of novel technologies within the clinical trial space, and particularly within clinical outcome meta-analysis via tools such as our Codex Clinical Outcome Database Explorer. As part of today's agenda, we'll start by exploring the overall clinical trial data landscape. We'll discuss the proliferation and value of unstructured content and some of the challenges associated with collecting, managing, searching, and ultimately extracting value from that type of data. We'll then jump into new approaches and applications for improving your exploration of this content and how it can be used to improve overall clinical trial success, from design to execution and evaluation towards the end. We'll also discuss some of the AI integrations that are being added into the Codex solution. I'll provide a brief demo, and then, as Sebastian mentioned, we'll leave some time for Q&A at the end, so welcome any comments or questions throughout the session as well. As we get started, it's important to understand the landscape and some of the challenges that the industry is facing. Currently, industry reports that only 1 in 5,000 drug candidates will result in a commercial product. And for each of these drugs, it takes an average of 10 years from discovery to approval to hit the market. And of that 10 years, 3 to 5 of those years are spent in discovery and preclinical stages. And while annual spending on clinical trials continues to rise, trial teams are feeling immense pressure to innovate faster while improving the safety and efficacy and overall success of those trials. One of the areas that we believe can improve those numbers and ultimately the overall trial success lies in the new and historical data that's being created by the industry, and particularly within the clinical trial lifecycle, on a regular basis. So during a clinical trial lifecycle, data is being collected from the early design phase to post-marketing and monitoring as well. And throughout that process, numerous documents and assets are being created throughout these milestones, and that can include everything from patient clinical data to eligibility screens and case reports. And all of this generated data informs the development of clinical trial summaries and that outline the successes and failures and other findings that may take place in that trial. And all of that becomes available for regulatory and public consumption. In addition to that, assuming the trial is successful, most teams will ultimately publish any study results in journals and other academic and industry publications. And finally, there's also additional reports and assets that are being developed as part of post-marketing to promote that new drug. All of that information holds incredibly valuable insight, but is really difficult to access and analyze, primarily because it's largely unstructured. In fact, nearly 80% of this data being generated is in an unstructured format. And what that means is it's predominantly language and text-based documents that take significant time and investment to actually analyze and screen and review to find the information that might be relevant for any new trial teams. And so traditionally, we've had to spend significant amounts of manual effort to analyze this content. But fortunately, there are new approaches that can improve these capabilities and also enable clinical trial teams to gain rich insights from these documents in a much faster and more accurate way. And that starts with clinical meta-analysis. And clinical meta-analysis is the process of extracting, combining, and normalizing clinical trial results. And this comes from a range of data sources that I was just outlining on the previous slide. And all of that data can be normalized and harmonized and brought together, and it can help trial teams draw conclusions about therapeutic effectiveness, improve overall trial outcomes, and also plan new studies. As part of Certara, and something that we've been focused on, is developing a expert team of curators who are focused specifically on this meta-analysis capability. In fact, to date, they've developed over 60 gold standard, high quality indication data sets that focus specifically on this meta-analysis data, and they're extracting it from all of that valuable content that's being created throughout the trial lifecycle. So for example, these data sets can include content from clinicaltrials.gov, company study synopses, regulatory documents, and other external communications such as press releases, conference posters, and investor communications that can help influence in our general understanding of its specific disease areas in clinical trial landscape. Now within that, we've focused most of our data sets into high-level therapeutic areas, and so currently each of these data sets focus on areas such as oncology, cardiovascular, central nervous system, immunology, and other areas as well. And within each of these data sets is content from over 10,000 different studies. And as I mentioned before, all of that is organized, augmented, and standardized for cross-study visualization and analysis. To add additional value to these data sets, we've also developed Codex. And Codex is a highly visual and web-based application that allows you to run clinical trial meta-analysis in a no-code platform. It stands for Clinical Outcome Database Explorer, and it allows you to gain overviews of specific therapeutic areas, covering common treatments and safety endpoints and efficacy endpoints as well. And then you can also connect via a data fabric your internal proprietary information with these rich indication data sets to expand your access to any of the clinical literature that you have available for your trial team. Ultimately, what this tool enables is your ability to reduce the time identifying and visualizing and running the analyses needed, particularly in phase 2 and phase 3 trials, to improve that drug safety and efficacy. Within the Codex platform are a number of visualizations. You can view summary level and study level views of your data, and I'll show some of that capability. In addition, you can do endpoint exploration and the ability to analyze time course treatments for specific drugs included in that database. You can also run pairwise meta-analyses and compare interventions across randomized controlled trials and to better understand how your drug needs to perform in the market. And lastly, you can also run network analyses as well, which enable you to identify and analyze relationships between other drugs in the market for that specific disease. Now, as I mentioned, as part of the Certara AI team, we've enabled a number of exciting deep learning and AI capabilities within the Codex solution. And that largely focuses on the leveraging of large language models. And these models are highly performant and enable you to gain rich context from complex literature. For example, all of those different types of clinical summaries and clinical study documents that we were discussing earlier on in the slides. From a AI perspective, we largely focus on two primary areas. One is named entity recognition, and that's the ability to identify key entities from structured and unstructured content, and that accelerates your insight discovery and is enabled by models identifying and highlighting specific terms of interest across those files. And secondly, and what we'll focus on a bit here in the demo portion of today's session, is in natural language question answering. What natural language question answering enables you to do is pose questions to your data and leverage models to scan through, identify, and extract certain answers that are available in the literature to accelerate your ability to find the information that is most pertinent for your research needs. Now, as part of exploring these AI capabilities, it's important to understand the capabilities of extractive AI, and particularly as AI and large language models have hit the mainstream industry discussions, particularly with the development of generative AI models such as ChatGPT. ChatGPT and other generative models, which you likely hear about regularly in the market, hold tremendous value and promise in the market. However, these vary differently from these extractive QA models that we're talking about here. And the reason for that is a generative model, like a ChatGPT, understands and trains on specific corpuses of information, but then ultimately creates and generates a novel answer based on information it has learned and been trained on. From an extractive QA capability, the difference is that you can pose a similar question to a model such as this, and instead of generating and creating a novel answer, it's going to go through your data and extract and deliver to you answers that it believes is true. In a use case such as Codex and clinical trial meta-analysis, it's critical that you can validate the origin of where those answers are found. And so having that reference material available to you through an extractive QA process is key to being able to validate that those answers are correct, and that the data that you're using to inform your trial overall is accurate and meets the needs and the standards for your specific use case. Within the Codex solution, we've taken these models and applied them into a low-code platform called Codex Curate. And Codex Curate is a AI smart spreadsheet technology that allows you to run these extractive QA models on a document-by-document basis. You can run these natural language queries, and with each question that you ask, Codex Curate is going to create a spreadsheet column with predicted answers that enables you to turn that unstructured data assets into structured data sets and structured insights that can be used to improve your overall indication database. One of the values within this, and what I'll show as part of the demo, is that not only do the models extract those answers for you, but we've enabled a number of human curation capabilities within the solution as well. And what that allows is the ability to extract and validate specific answers and where they've been found in the literature. So again, it allows you to have that reference information to ensure that the data that you're using to feed into Codex is validated and accurate for your given needs. One of the additional benefits related to this solution is what we call our reference data fabric. And this expands your organization's capability to not only access the rich indication data sets that are being provided within Codex, but then you can also expand to additional publicly available information from a series of biomedical literature resources. And so within our reference data fabric, we provide a deep learning analyzed index of resources from PubMed and PubChem, the preprints, clinicaltrials.gov, and other biomedical resources as well. This can integrate directly into the Codex visualization and the Codex Curate tool. And one of the reasons that I'm showing this as well is because as part of the demo, we'll actually be extracting and creating a novel data set based on content from this reference data fabric as well. So at this point, I'm just going to switch screens momentarily and jump into a demo. Okay, So within the Codex application, you are able to run a number of different analyses, which I will show next. As I mentioned before, the Codex solution is a highly flexible web-based application and is highly responsive and includes a number of different intuitive and no-code visualization tools. Within the solution, I have the ability to select specific indication databases that I want available for my given research task. And then when I choose a specific database for exploration, I gain a bird's eye view and a dashboard view of all of the assets that make up that database. So I can see the number of references and study arms and attributes specific to that database. I can also see things such as common efficacy endpoints existing within this database, safety endpoints, and then I can also see some of the evidence characteristics that make up this database as well. So I can see the number of phase 3 trials that are included, the number of blinded trials, and also the randomized clinical trials that are included in this database as well. Beyond having a top-level view of this database, I also have the ability to dive into the specific aspects of that database, both from the database as a whole, where I can see the various attributes at a summary level, as well as view specific references within the database and other outcomes within the database and view that information, as well as the ability to dive into by study level into the specific assets that make up that database itself as well. So again, I get a similar summary level of the study, and then I can also view additional assets that are being extracted from that study and where that's existing within the database as well. And I have the ability to switch studies based on the specific information that I might be trying to investigate and look at as well. For this demo, what we're going to focus on though is the ability to analyze against specific drugs within that database. And so within the solution, perhaps I want to filter down for a specific drug of interest to see how it's performed in other trials and see what some of the attributes exist within the clinical trial landscape. So I can choose and filter down my database, and perhaps I want to focus on a specific randomized drug. I can select from the database any of the drugs that have been listed, and I can see a tally of mentions within the database of that specific drug. For the purposes of this demo, I'm going to focus on a pretty common drug, fingolimod, which is also known by the market term Kesimpta as well. And I'm going to add that filter here. And so now I can see, based on the selected filter, exactly how many studies and references and study arms exist related to this specific drug that's been identified. And again, I can see my dashboard updating in real time based on the filters that I've added to this view as well. From there, I likely will want to do some additional exploration of the tool itself, or of that drug itself, as I should mention. So perhaps I want to analyze the time course study for that specific drug, and I can see various charts related to those studies and see how that drug has performed. And I have different filters for that as well. I can also view time course by treatment against those various studies and analyze that as well. Beyond that, I may want to analyze and understand how this drug has reacted in specific trials, and perhaps I want to investigate adverse events related to that specific drug. So in that case, I could use my pairwise meta-analysis, and perhaps I want to focus on things such as bronchitis as an adverse effect. I can analyze exactly how that has reacted in certain studies, and I can see from each study sort of the various effects that have been reported. I can see the average effect by the dotted line, And then you can also see the variability and average of those effects in general based on this blue line at the bottom, as well as the diamond within the plot itself. As I run this analysis, I have lots of flexibility in the platform itself to expand the visualization of this as well. So I can adjust my columns within the graph itself. And so I can add any additional filters here. And I can also do this to my other columns as well. And this will change my visualization based on the given needs that I have for the specific analysis that I'm running. Once I'm happy with the overall filtering and the results, I then have a summary level overview of the specific analysis that I've run, and I can see where the data is coming from, as well as the analysis set. And then Codex also provides me with a summary report and data download that I can export from the system and deliver across to my other members of my team and for this clinical study, so that they can also review and analyze any of the insights that I've pulled from this. In addition to that, I can also analyze all of the drugs in the database and view their relationship within that as well. So in this case, I can build out a network plot as Codex begins to generate that of all of the drugs that exist within this database and see their various relationships with each other. I can also zoom into specific areas. So I was looking at fingolimod before. Perhaps I want to just investigate this specific area of my network meta-analysis, and I can view the various relationships that exist with other drugs in this database as they relate to fingolimod themselves. And again, I have various ways that I can filter this network plot, as well as being able to add specific filters if I want to run specific analysis, say against a finite number of drugs within the database that I want to be able to understand their relationship. And again, similar to the pairwise meta-analysis, I still have the ability to review summary level information as well, and data selection overviews related to that too. So there's a number of flexible ways in terms of being able to understand within Codex the way certain drugs perform throughout the clinical trial landscape, and that ability to understand how the drug that you may be focused on in your trial needs to improve its performance in the way it needs to perform overall if it's going to enter the market itself. Now, as I was mentioning, as I started to run through Codex and from the study level, you can see that there are certain attributes that are being extracted within these summary levels, and then within the various details of the study data set as well. Now, as I mentioned, part of the value is that ability to also connect in your own internal data and also run additional extraction from your proprietary or internal data that might relate to a specific trial that you're running or a specific drug or indication that you're studying. And so the Codex Curate solution enables you to do just that. And so within this tool, it is, as a reminder, a smart spreadsheet tool that connects directly into Codex and allows you to run both metadata extraction from sets of documents and then also those natural language question answering on a document by document basis to begin to build out similar data sets that can be fed into the Codex platform. So in this case, perhaps I'm following the same Codex analysis I was doing before, and I want to pull in additional documents from my internal data related to multiple sclerosis. I can search for that information and whatever data source it might be coming from. In this case, I'm using that reference data fabric, so I'm pulling from clinicaltrials.gov, but this could also be any other data base that you may want to connect into or specific document sets that you have. And then I can run a variety of different text extraction functions within the tool itself. So perhaps there's specific metadata properties that I know exist within these trials that I want available in my data set. Maybe I want to know the lead sponsor. Maybe I want to know the interventions within those trials, and I want to know the age list for that trial itself. I can request that information and have the models pull that out for me, as well as sections of the document as well. So maybe I want to know the study design. By clicking Save, the Curate tool is going to extract that information and deliver a clean spreadsheet and structured spreadsheet with those specific insights that I've requested the models to identify and provide for me. And now I can see that specific information already built out into a spreadsheet for me, including larger sections of text like a study design that can be extracted for me and available within a specific spreadsheet cell. Now within this, I also have the ability to view source documentation as well and see those documents to do further analysis on them. As I was mentioning, one of the values is still in that natural language question answering. And at some point within your spreadsheet creation, you're likely going to want to identify insights that aren't part of the document metadata. And that's where those natural language models and those large language models come into play. So perhaps I want to ask a couple of questions of these documents and have them begin to build out the spreadsheet further for me. So maybe I want to know the age range of the patients, any of the study arms mentioned, any of the drug that are mentioned, and the outcomes that are being measured in that study. What these models are going to be able to do is understand that question and scan each of the documents within my document set and deliver predicted answers for me that I can then view. So as I run the solution, I begin to see the model go through and identify specific answers that are being found. And then I can begin to analyze the various predictions that have been identified within my spreadsheet. So now I can see specific answers that have been found, and I can go in and view exact answers that have been identified within the document sets themselves. So you can see in a question like this, what drug is being mentioned. There are multiple drugs that are mentioned within this document, and I can go in and validate based on the confidence score that the model has given me, or by viewing the document itself, either from an evidence view or accessing the full document to say, yes, I believe that this is a correct answer for my given use case, or no, there's an additional answer that I want. And by validating that, you're able to fine-tune this data set so that it meets the specific needs and parameters that are required for your meta-analysis data set. And so these document sets can be shared with teams of curators who can go through that validation and ensure the accuracy and of that meta-analysis data that can then be fed into Codex via our data fabric and harmonized with those additional data sets so that you can gain not only a view of that gold standard expertly curated data sets that our Satara team can provide, but then also the analyze that across and against your internal documents that may hold very similar insights as well. So after it's sort of a brief demo of the Codex solution itself, I thought it would be useful just to provide sort of an overview of how the solution is being used with customers today. And so unsurprisingly, and what we've talked about a lot is the challenges and the problems that some of our users are trying to solve. And that revolves around both that difficulty in analyzing literature-based research and a need to have those data-backed insights to improve go, no-go decisions, particularly around those phase 2 and phase 3 trials. What you need to ultimately improve that are things that are included within the Codex solution. So having a robust library of clinical outcome databases that you can review and analyze, having highly visual and intuitive tools for running those various analyses, and then having the ability to connect in your own internal data as well. And that can provide a number of different benefits for clinical trial teams. It can improve overall trial design and trial simulation. And then it can also better inform and provide the data needed to better predict and understand go and no-go decisions for that trial moving forward. As part of Codex, there are a number of valuable takeaways from the solution. That ability to have data accessibility and allowing to integrate both expertly curated data sets with publicly available information in your own internal clinical outcome data into a single solution. Having those expertly curated data sets that are being developed by PhD level data curators and researchers from Certara, accelerating your team's ability to have that data available to them instantly. Accelerating the research and actual data identification through those large language models, and then being able to improve that data set curation with your own internal data as well. We will be showcasing a number of our solutions both at HIMSS next week in Chicago, and then also at BioIT World in early May in Boston. At BioIT World, we'll also be speaking on overall AI approaches for clinical outcome meta-analysis, including an expansion of the Codex solution overall that we discussed today, as well as our other analytics tools across the drug development lifecycle. And so with that, I'll pause now and leave some time for Q&A.
Yeah, thank you, Nick, for that great presentation. Very, very insightful. As a reminder, please submit your questions through the chat pane, and we will address them. A couple of questions already came through during Nick's presentation, and let's start with this one. Question is, how does my internal data get fed into this platform?
Oh, sure. That's a great question. Happy to answer it. So as part of the Codex solution is a flexible and secure data fabric on the back end. And what that data fabric has is it features a number of data connections that enable deep learning models to index and analyze any of your connected data sources. And that content, that index of information becomes searchable and accessible and visualized in the Codex platform. So there's a flexible data fabric on the back end that enables much of that capability.
All right, great, thank you. Another great question. How often is the data updated as new research comes to the market?
Sure. Yeah, we get this question a lot. So in terms of the Codex indication databases that I was showing earlier, those are updated on a regular basis, and that indication data, those Codex curators and researchers are analyzing and building out and releasing new versions of those data sets on a regular cadence. In addition to that, any of your data that you connect into the solution can be re-indexed on a regular basis and become available to you on a daily outcome. And so, for example, that reference data fabric that I was introducing earlier, of those publicly available biomedical sources, those are updated on a daily basis to ensure that you as an organization have access to the latest publications and information that is being published on it as it becomes available.
All right, thank you. Thank you for that answer, Nick. And we have one more question that came through, and the question is, can you share an example of this AI being used by a customer?
Absolutely, yeah. So we typically see our customers coming to us to analyze difficult to access and apply these models against unstructured data. And so, whether that be some of the clinical study data that we were talking about today, or additional data assets that they might have available, for example, semi-structured CRO reports and things of that nature. And so, by using some of the AI and large language models that exist in a platform like this, they can extract those capabilities, those data insights much faster, as we were showing. And ultimately, that provides a number of different efficiencies for our customers. So it can improve the productivity of overall research teams, and we've seen productivity increase of over 25%. In addition to that, it also improves data curation and data collection workflows, usually around a 30% improvement based on manual processes as well. And we've seen most of our models reach an F1 score in the high 90s in terms of their accuracy and being able to identify and extract the information that our users are leveraging those models to identify.